I haven't given you my panel over here because we're in the process of validating it, but it's a 52 gene panel, 3 genes with entire coding region targets lung, colon, thyroid, GI cancers, gliomas, melanomas, and GISTs. And some data that I'm showing you in the panel on the right is looking at about 16 to 20 samples. We've run a lot more since then. These are genes that were not really too many of them present on the previous panel that I showed you. I've included genes like TERT, promoter mutations in the TERT gene. Those are present in glioma, and those actually have prognostic significance. So those are included in the panel. GNAS and GNA11, which are present in uveal melanomas, those are really indicative of diagnostic for uveal melanomas. Those are included in the panel. What else have we besides the other ones? We have done a whole lot of genes over here. I'm sorry, I cannot see all those genes written over there, but these are not genes that are present in a lot of the run-of-the-mill hotspot panels that you see. In the second column, what you see over there is actually the observed allelic fraction that we got. These were all samples that were run on one of our Columbia panels earlier, so we knew what mutation to expect, and we checked those mutations against the mutations that we got here. In the column after that, I want you to see the coverage. Look at the coverage that we obtained in this panel. Remember, these are 22 samples run on a V2 flow cell. You can actually run, we even run up to 26 samples on a V2 flow cell. It's a pretty large panel, 52 genes with 3 genes, whole coding region. So it's about a little over 200 kb actually in size. The allelic fractions that we got in the last column varied from 3% all the way up to 80%. And the data we got, we liked what we got, so we've decided to actually go ahead with the New York State submission